Hi friends, it's me Anila KF, first year MSc Biology, St. Mary's College, Thrissur. Today's topic, structure and function of different class of immunoglobulin. Immunoglobulin are also known as antibody. They are glycoprotein molecule produced by plasma cells. They act as critical part of immune response by specifically recognizing and binding to particular antigens such as bacteria or virus and aiding in their destruction. This is structure of immunoglobulin. Five classes of immunoglobulins are IgG, IgM, IgA, IgD, and IgE. These are distinguished by the type of heavy chain found in the molecule. In IgG, gamma heavy chain, IgA, alpha heavy chain, in IgM, mu heavy chain, in IgE, epsilon heavy chain, in IgD, delta heavy chain. IgG, most abundant class in serum, constitute about 80% of total serum immunoglobulin. It is a monomer, molecular weight 150 kilodalton, half-life 23 days. This is the structure of IgG. It's containing 4 domains and hinge region, distributed equally between intravascular and extravascular. IgG1, IgG2, IgG3, and IgG4 are the four subclasses. Structural characteristics that distinguish the subclasses from one another are the size of hinge region and number and position of interchain disulfide bonds between heavy chain. Structure of IgG subclasses. Human IgG3 has 11 interchain disulfide bond. IgG1, IgG3, IgG4 cross placenta and protecting the developing fetus. IgG3 is most effective complement activator, followed by IgG1. IgG2 is less efficient. IgG4 is not able to activate complement. IgG1 and IgG3 bind with high affinity to Fc receptor on the phagocytic cell, mediate the opsonization. IgG4 has an intermediate affinity for Fc receptor. IgG2 has extremely low affinity. IgGM 5% to 10% of total serum immunoglobulin. Its half-life 5 days. Its pentamer secreted by plasma cells. Monomeric IgM expressed as membrane-bound antibody on B cells. First immunoglobulin class produced in a primary response. Is containing additional Fc-linked polypeptide. It's called J chain. It's required for the polymerization of monomer. It's added again just before the secretion of pentamer. It has 10 antigen binding site. It can bind 10 small hapten molecules. Because of steric hindrance, only 5 or fewer molecules of large antigen can be bound simultaneously. This is structure of IgM. Complement activation required to Fc region. Pentameric structure of single molecule of IgM fulfills this requirement. Its large size does not diffuse well, therefore it is found in very low concentration in the intercellular tissue fluid. Next is IgA, constituting 10% to 50% of the total immunoglobulins in serum. It's present in breast milk, saliva, tears, and mucus of bronchial, genitorial, and digestive tract. Half-life 6 to 8 days. IgA occur in two forms, serum IgA and secretory IgA. Secretory IgA containing glycine-rich polypeptide called secretory component. It protects IgA from denaturation by bacterial protease. IgA does not fix complement but can activate the alternative complement pathway. It promotes phagocytosis and the intercellular killing of microorganisms. IgA1 and IgA2 are the subclasses of IgA. This is the structure of IgA. Next is IgE. Half-life 2 days. It's heat labile. It's mediate PK reaction. It's also mediate hypersensitivity reaction that responsible for the symptoms of hay fever, asthma, hives, and anaphylactic shock. This is structure of IgE. It has 5 domains. Last one is IgD. This constitutes about 0.2% of total immunoglobulin serum. IgD was first discovered when a patient developed a multiple myeloma whose myeloma protein failed to react with the anti-isotype antisera against the known isotype IgA, IgM, and IgG. Its half-life 2 days. This is the structure of IgD. There are 4 domains. IgD together with IgM is a major membrane-bound immunoglobulin expressed by mature B cells. Its role in physiology of B cell is under investigation. No biological effector functions has been identified for IgD. Reference. Thank you.